Yay! I'm excited for this segment of this lecture because we get to talk about my favorite of the four compounds of life, proteins. Yay! Proteins are awesome because they are the most diverse molecule out there. They're so simple in their structure, yet so incredibly diverse and complex in their function. And you know how they say everything you needed to know you learned in kindergarten? Well, you learned about shapes, and proteins are all about their shape. You change their shape, you change their function. So we're going to start off by talking about some of the amazing, different, and varied functions that proteins serve, and then get into the biochemistry, talking about what makes them up, and how do these amazing molecules fit into the secret of life, i.e. the central dogma of biology. So we'll spend the entire term talking about proteins because they play a role in pretty much everything we do. So we're going to spend a lot of time talking about proteins. When in doubt, it's probably a protein. So let's get started. So again, the most diverse cell compounds that there are. They come in all sorts of shapes and sizes. So we've already mentioned how proteins can serve as enzymes. So now keep this in mind. All enzymes are proteins, but not all proteins are enzymes. Got that? So enzymes serve actually themselves a couple different functions. They serve as catalysts, as we mentioned before. In other words, it is the enzymes that allow, I'd say, most of the reactions of the body to occur. In order to go from reactants to products, without the help of enzymes to speed up the reactions, it could take billions of years for that reaction to occur. And most of us don't live billions of years, so that wouldn't work. But thanks to enzymes, that same reaction can occur in a fraction of a second. So how do enzymes do this? We'll spend more time later in the term talking about the nitty-gritties of enzyme activity. But in general, they lower the activation energy of metabolic reactions. So in other words, to go from reactants to products, it takes a certain amount of energy. You got to get over an energy hump. It's kind of like to get to the other side of the mountain. You got to expend some energy to climb that mountain. Well, the amount of time it would take to get up that mountain on its own would be, as we said, billions of years. And so what the enzymes do is they lower that activation energy. They kind of shorten the mountain, so it doesn't take as much energy to get to reactants, from reactants to products, and thus the reaction can occur and life can go on. So they serve as enzymes. Enzymes also help to break things down, as we saw before, so they hydrolyze bonds. Proteins, many proteins serve structural roles. So of course we have our hair and our nails, fur, it's made of the same proteins that our hair is. But also things like cartilage and bones, the feathers of birds. So lots of different structural roles, and we'll talk some more examples about that in a bit. They play a very important role in cell communication. So we've already talked about lipids and how some lipids can serve as hormones for cell communication, but so can proteins. And so we'll give some examples of that as well. They store things like amino acids. They serve a defensive function, i.e. immune function for our bodies. They serve for movement and transport. In general, they just do all sorts of things. They are so cool. But no matter whether you're a structural protein, an enzyme, an immune protein, whatever, all proteins of all organisms on Earth, whether a human, a tree, an alga, a fish, a bacterial cell, etc., 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 all the proteins on Earth are made of just different combinations of the same 20 amino acids. So the monomers of protein polymers are amino acids. And this is very, very important because this feeds into the secret of life. And we're going to spend quite a bit of time in this term talking about how do we go from our genetic code, DNA, to a chain of amino acids to the proteins that carry out specific trait functions. Very, very cool. Again, the key is, like so many things we've been talking about in this lecture, the beauty of life, the diversity of life is all predicated on very simple things and very few things. And so in this case, 20 amino acids, all made of the same basic elements of the periodic table, chomps. So let's see some examples of these functions in action. So these two little diagrams just summarize what we just said about some of the functions of proteins and give examples. So we said we can have enzymatic proteins. selective acceleration of chemical reactions. And so we're going to spend more time later talking about how this happens. But in general, an enzyme is kind of like a matchmaker, and it's going to allow two substrates, i.e. two reactants that need to come together, will fit into this matchmaker. Maybe they would have never come together on their own, but the enzyme matchmaker fits with them perfectly and gets them together, and then you can get your product. Defensive proteins, things like antibodies, which you've probably heard of. If you're exposed to a particular disease, you may build up antibodies so that next time you get exposed to that disease, you won't get it. Storage proteins, for example, oval albumin, you might be familiar with. Maybe you had some for breakfast. That is egg white. Believe it or not, oval albumin, the egg white, was not built for you to eat. It was built as a storage for amino acids for the developing chick, the embryo. Transport proteins, we already said hemoglobin is an example of that. So many different transport proteins in the body, and we'll be encountering some of those this term. Hormonal proteins, for example, insulin is produced by the pancreas, and that helps regulate our blood sugar levels. Muscle, so we have lots of proteins in our muscles. So for example, actin and myosin, which are the protein filaments that slide past each other to allow muscle contraction. I have no muscles. Receptor proteins, so this is important for cell communication. We're going to spend a whole lecture on talking about cell communication because it's fascinating. But for example, if I wanted to contract my muscle, I have to have something connected to my nervous system to tell my muscle to contract. And thanks to proteins, we're able to get signals from our nerve cells to our muscle cells. And of course, we talked about structural proteins like keratin, which is what makes up our hair. So those are just some of the beautiful things that these proteins can do. So it's all about the function. Now, on the next slide, I'm going to show you all of life on Earth. All of life on Earth on one slide, because it's those 20 amino acids we talked about. So again, the protein monomers are amino acids, and there's only 20 of them that are produced by life forms. So this is pretty amazing. And remember how we said the shape of the protein determines its function, and its shape is determined by the order of amino acid monomers that make it up. And so I just think the amino acids are so fascinating because even though there's only 20 of them, they produce this diversity of proteins, yet the amino acids themselves, those 20 amino acids, are so similar to each other. And they're all made of the same few elements of the periodic table: carbon, hydrogen, oxygen, nitrogen, sulfur. So it's really, really amazing. So let's take a little bit closer look at these amino acids. Now, if you are going to take biochemistry at some point, you probably will have to actually memorize the actual chemical structures of all 20 amino acids. I'm not going to make you do that right now, but it's very interesting to look deeply at them just to gain an appreciation for life and how could life evolve. Life could never have existed without these amino acids, yet they're so fundamentally similar to each other, yet slight differences are the difference between a functional protein and a non-functional protein. So let's take a look. Notice that these have actually been spread out on this table in a particular order. They're divided by nonpolar amino acids, polar amino acids, and electrically charged amino acids. Now, if you look closely, notice that all of these amino acids, no matter whether they're polar, nonpolar, charged, or what, they all have the same basic backbone. So they basically have a nice little carbon there in the middle with a hydrogen coming off the top. So carbon, hydrogen, an amino group off on one end, an H2. So remember your functional groups, there's an amino group, and hence the term amino acid. And on the other end, we have C double bond O, OH, Cu, right? You remember that functional group? Carboxylic acid. Awesome. So we have a carboxyl group on one end, an amino group on the other, a hydrogen, and all these guys are connected together with what we call the alpha carbon. Now, the rest of the stuff, the little tails that come off of here, we in general can call all these tails R groups. And these are also called side chains. So these are side chain R groups. And that is what gives the amino acid its individual properties. And when you put these amino acids together to make the polymer protein, that's what gives the protein its function. And so we're going to spend a lot of time later in this term talking about mutations and what happens when the DNA gets mutated and that codes for the wrong amino acid. You get the wrong amino acid, i.e. the wrong R group, you actually can change the properties of the protein and change the trait. Maybe you don't get the trait at all. Maybe you get the wrong trait that could cause a genetic disorder. Or maybe you get a slightly different trait that confers an advantage to the organism, and that can help lead to evolutionary change, natural selection. So let's take a look at how similar these are. These are so cool. So check out glycine here. That's the simplest amino acid. It's nonpolar. And its neighbor here, alanine. Notice that each amino acid has a 3-letter abbreviation. It also has a 1-letter abbreviation, but that's not shown on this chart. Notice the only difference between glycine and alanine is that glycine only has a hydrogen as its R group. Alanine has a methyl group. What's the chance that in evolutionary time, in chemical evolution, you got a slight chemical change and a hydrogen got replaced by a methyl group, and boom, you have a brand new amino acid? Not that difficult if you think about it, and in fact, we can make these things happen very easily in the laboratory. Check out the difference between alanine and valine. You have 2 methyl groups down here and a CH group there, or between valine and leucine, just differ by CH2 up here. So really interesting that some of these amino acids are very similar in size, shape, and properties, and others are very different. And so the severity of a mutation depends on what kind of amino acid change you got. So this will make more sense later on in a semester when we get to what's called protein synthesis. But for example, let's say the DNA was supposed to code for making, a certain segment of DNA was supposed to code for making the amino acid, I don't know, let's say methionine that you see right here. Okay, this is what we call the start codon in protein synthesis. Let's say it was supposed to make that. Well, we know that methionine is a nonpolar amino acid, and it's a fairly large amino acid. It has this big R group here. Let's say that the DNA mutated, and instead of methionine, you get tyrosine being produced. Well, you can see tyrosine is a very different amino acid than methionine. It has this big, what we call a benzene ring here. It's polar instead of nonpolar. And so this can cause a big change in the formation of a protein. Remember that opposites attract, right? And so if you had a nonpolar amino acid and it was supposed to match up with, say, another nonpolar amino acid or a polar amino acid, and you changed the polarity of that amino acid through mutation, all of a sudden, what do these guys want to do? They want to repel each other. That changes the whole shape of the protein, and it likely will lose its function or at least have a very altered function. So this is very important. And so I really would love for you to really get a feel of which amino acids are nonpolar, which are polar, which are acidic or basic, i.e. negatively charged are the acidic ones, these two. And the positively charged ones are these basic ones that we see here, these 3. So if you get a feel for this, it'll give you an appreciation later on when we study genetic disorders and evolution. So, but it's these amino acids and their properties that are the basis for evolution of life on Earth. It's very deep. And that happens to be my favorite topic in the world. Okay, so you get the idea. So we said for carbohydrates, the monomers were monosaccharides. For lipids, they don't have true monomers, but they're built of glycerols and fatty acids. And in the case of steroids, they have a 4-ring backbone. And for proteins, the monomers are amino acids. And we haven't gotten to nucleic acids yet, but we'll be doing that soon. Okay, so we know proteins are made up of amino acid monomers. Now, when you get these amino acid monomers put together, we call that a polypeptide chain. And so we can say polypeptide polymers are made of amino acid monomers. And so we have a diagram here that's just going to show you how we form this polypeptide chain. So here is one amino acid, here's another one. Let's say we wanted to put these guys together to form part of this chain. What kind of reaction are we going to use? Dun, dun, dun. Dehydration synthesis reaction, right? And so basically on the carboxyl group of one amino acid, we're going to take off a hydroxyl group, and we're going to take a hydrogen off the amino group on the next amino acid, H2O, boom, and now we have water as a byproduct. And we have now put these two amino acids together, forming what's called a peptide bond. And so you can also look at this polypeptide chain and realize that the amino group on one end is called the N-terminus. So over here in our actual chain we're building, this would be your N-terminus over here. And the carboxyl side would be your C-terminus. C for carboxyl, N for our amino, N, well, anyway, amino, think of that with an N sound to it. Anyway, here's an actual ribbon diagram of a beautiful protein consisting of multiple polypeptide chains. So again, this is just reminding you that a peptide is a chain of amino acids. So the amino group here is your N-terminus. These different, it looks like we have 1, 2, 3, 4 amino acids joined together here via peptide bonds represented by these lines. And at this carboxyl end, that's your C-terminus. So that's a polypeptide chain. So that's how it's done. Just like everything, when we want to make a polymer, we're going to use dehydration synthesis to join the monomers. And in this case, we're joining them via peptide bonds. So there are, as you just saw on the previous slide, different ways of representing what a protein looks like. And these are really cool. Often they're generated by computer modeling. And they're just really cool. They're just different ways of representing the same thing depending on what it is you're trying to show your viewer. Now, just like we talked about before, in reality, molecules look more like this space-filling model right here. So this would be a space-filling model of a protein, whichever protein it happens to be here. It's three-dimensional. Notice here there's a groove. So this is very important to many proteins, that they have a groove where another molecule can come in and fit like a glove, or like you're doing a jigsaw puzzle, fit right in there. And often, once that other molecule attaches into that groove, it causes what's called a conformational change of that protein, and that actually changes the protein's function at that point. It can do something else. It becomes active. So we're going to spend a lot of time talking about that. A lot of life is about molecules attaching to proteins, causing conformational changes, basically changing the shape around and causing a new activity. So that same protein can be represented in a wireframe model. This is basically each of these little parts here would be a different amino acid. So it allows you to see the individual amino acids in a little more detail, where you're, well, it's kind of a combination between a ribbon diagram and a wireframe. They're sort of stuck together here, but we'll call both of these wireframes because that's the base of it. And then here's your ribbon model. And so the ribbon models, there's different versions of them. The idea is it's showing you the different shapes of the different polypeptides that make that protein up. So we'll talk about shape in just a second. So again, proteins' functions are dependent on the shapes. And so you alter the shape by changing the amino acid sequence, and you can change the function of the protein. So it's all about shape. And so a straight polypeptide chain, or just a polypeptide chain that hasn't folded up into a true protein, we call this the primary structure. So for example, here's your N-terminus, and here's your chain of amino acids joined by peptide bonds. And notice it's just, they're just hanging out. They're just in a straight chain. So that's a primary structure. And so many larger proteins are made up of several different polypeptide chains that are in their primary structure. If we go over to the secondary structure here, we have two possibilities. We have what's called an alpha helix, and this is where the configuration of the amino acids causes the polypeptide chain to turn into a spiral, into a helix. So that's an alpha helix. So on the previous slide, remember there was like these little barrels as part of that ribbon thing. So that would be representing alpha helices. Another secondary structure is what's called a beta pleated sheet. And this is where the configuration of polypeptide chains actually forms a pleated sheet, a pleated formation. Now you get several primary and or secondary structures together you can form what's called a tertiary structure. This is kind of a globular, just blob of polypeptide chains. So that is one shape to a three-dimensional protein, is a tertiary structure. And why does it do this? Why do these chains fold up on each other and get it into a nice blob? It has to do with the interactions between the R groups of the amino acids. So remember how some are polar, some are nonpolar, some are negatively charged, some are positively charged. So obviously if you have a negatively charged amino acid here and a positively charged amino acid here, those guys attract each other, right? Opposites attract, and that causes those chains to kind of come together and form because of the force that's there, or the electrical force. So that causes the folding into the tertiary structure. In fact, if we look closer over here, you can kind of see where this is coming from, like different ways that these different R groups from the amino acids attract each other. So you've already learned about van der Waals forces. Remember, those are really weak forces, but when taken over a large area, they actually become a strong force. So you can have van der Waals interactions between R groups, hydrogen bonds. In the case of some, you have what are called disulfide bonds, where sulfur on this amino acid and on that form a bond together, called a disulfide bond. Ionic bonds, which you guys are well familiar with. So lots of different ways that these guys will attract each other, and that causes the whole chain to fold up and make a little glob. Now again, you change the amino acid. Imagine I change this amino acid here to the opposite charge. Now instead of coming together, those guys want to repel each other. That unfolds the protein, changes, or makes it lose its function. And finally, when you get several of these tertiary structures together, or many different polypeptide chains, each with kind of its own shape, when they come together into one big structure, we call that a quaternary structure, multiple polypeptide chains. So for example, hemoglobin is a quaternary structure. It has actually 4 different groups to it. Here is just another picture showing you computer-generated models of these different structures. So going over here, here is the secondary structure. You can see this is your alpha helix right here. Here's another secondary structure, your beta sheet. Isn't this cool? That's so pretty. I would hang these on my wall, definitely. Here's a tertiary structure, so you can see the representation of these different polypeptide chains here. So for example, that one's more of a primary structure polypeptide chain. There's an alpha helix right there and right there, etc. So, so the more complicated the polypeptide chains, the more interesting looking the model is. And finally, when we take a bunch of these guys together, you can get a quaternary structure. Okay, now, how do we get these proteins to fold up the way they're supposed to go? So, you know, these forces between our groups make them desire to fold up to each other, you know, in a certain way. But usually, especially if it's a large protein, it's going to need some help. And so when you learn about protein synthesis, you're going to learn what happens at each stage. At the final stage, right before the polypeptide chain is ready to fold up into a protein, it will meet another protein. So when in doubt, just say proteins. Proteins do everything, even help other proteins. There's one kind of set of proteins called chaperonins, or I like to call them chaperones. So chaperonin proteins literally chaperone the forming protein. So imagine when you were a kid and you went on a field trip and they always put a chaperone on the bus to kind of help the kids stay in formation and structure and keep you from running off. Well, that's what these things do. These chaperonins chaperone the forming protein. So let's say, you know, protein synthesis has occurred. We've created this beautiful primary structure polypeptide chain of all these amino acids. Well, before this guy is done, it's actually literally going to go inside of a chaperonin protein. Literally, this guy has a cap to him that comes off. The polypeptide chain goes in. Like we said before, that's a molecule attaching to a protein molecule, causing a conformational change due to changes in polarity and things like that. And that conformational change will actually, it's kind of like, okay, get it folded up in its right position, and cap comes back off, and boom, this correctly folded protein leaves. So this chaperonin is kind of like, you know, it's what's going to lead to the protein forming in the correct way. And here's an actual computer-generated model of what a chaperonin protein really looks like. And you can see it itself has different sections to it. So each section of a large quaternary protein we call a domain. So in this particular case, this guy has, looks like 4 different domains that make up the chaperonin protein. And he's produced the same way as any other protein. So the chaperonin protein would have needed chaperonin proteins to become a chaperonin protein. Cool, huh? So just bottom line, 3D structure determines a protein's function, and it gets help from chaperonin protein. So by now it's probably been hammered into your head why the 3D structure of a protein is so vital to its function. Any little change, or especially if it's a big change, in that structure, in that shape of the protein can cause the whole protein to change shape and therefore lose or severely alter its function. So let's look a little bit deeper at our example of hemoglobin. So recall, hemoglobin is a transport protein. It carries oxygen throughout our body, and thus it's very important, right? Without hemoglobin, you can't breathe, and your cells can't breathe, so that's really important. But let's look at what goes on when a slight mutation in the genetic code, the DNA, that codes for the order of amino acids to create the polypeptide chains that make up hemoglobin. What happens if a slightest change, just a little itty bitty change, and we'll be talking about the structure of DNA so you can learn what that change is. What if it just alters 1 amino acid out of the 287 amino acids that make up this big quaternary structure protein? What if you just altered 1? And particularly amino acid number 6 of the 287, what does that do to the overall structure and function of the protein? And ultimately, how does that alter a person? So this will give you an idea of how genetic disorders can occur. So again, hemoglobin, which we see up here, consists of 287 amino acids broken into 4 domains or subunits. And actually, if you look at the quaternary structure here, so this would be a normal production of hemoglobin. There are 4 subunits, 4 domains. We have 2 alpha subunits and 2 beta subunits. And so this is showing you, what the normal shape of one of these beta subunits would be. Now, the order of amino acids that make up all these subunits are such. So again, there's 287 of them. This diagram shows you the first 7. And what we're going to do is pay attention to amino acid number 6. So in a normal person who does not have sickle cell, amino acid number 6 in the chain would be glutamic acid. And if they get the correct order of all these 287 amino acids, they get the production of a nice, beautiful, donut-shaped red blood cell. This is what beautiful blood cells are supposed to look like. They're capable of carrying oxygen and flowing through our blood vessels and doing what they're supposed to do, and thus we do not have sickle cell anemia. But somebody who has sickle cell means that they were born with a mutation at amino acid number 6 of the 287. Instead of their DNA coding for the production of glutamic acid at number 6, it codes for the amino acid valine. So 287 amino acids, just one, just number 6, has the wrong amino acid. And what that ends up doing is altering the beta subunits of the hemoglobin protein. And you can see it's altered its shape. And by altering that shape, getting this little divot in those subunits, it actually causes these proteins, the hemoglobin proteins, instead of being nice and separated from each other like you see up here, they start aggregating. And that causes the cell itself to sickle up, as you see here, the red blood cell, and therefore it can't get the proper binding of oxygen that it's supposed to have. And so someone with sickle cell, if it's untreated, can die very easily, a very, very painful death from sickle cell anemia. So let's take a deeper look at amino acid number 6. What you're supposed to get, glutamate or glutamic acid, same thing, versus the mutation valine. So if we go back to our list of amino acids here, so remember there's only 20 that make up all these proteins. And if we find valine, so here's the amino acid valine right here, and here's glutamic acid. So right off the bat, you can see they differ in terms of their size. So valine is kind of a short R group right here, right? A CH and two methyl groups, whereas glutamic acid is quite large. Notice that glutamic acid is negatively charged. It contains a charge, whereas valine is nonpolar. So right there, you can see that you've changed the polarity of the amino acid at amino acid number 6 of the hemoglobin, and this is going to cause a conformational change of that beta subunit. And the result is a divot in the shape, which alters the function, which is the ability of that whole protein to bind to oxygen. So very interesting how just little itty bitty changes in our genetic code and our amino acid sequence can cause huge changes at what we call the phenotypic level, the expression of the protein, the expression of the trait. So we'll be getting a lot more into this when we get into genetics and molecular biology and evolution. So I hope you have enjoyed this discussion of proteins. I know I have because I love them. They're so cool. And it doesn't end here because these are themes that will carry throughout the course. So we have one more compound of life to go, and that is getting to the fundamental, and that is nucleic acids, which include our genetic material, including DNA. So let's go on to nucleic acids.